Should I start?
Yes, ma'am.
Yes. So, good evening, everyone. As we have started the GIT series, so today is class 7, and the topic we have is small intestine. So before starting, let's discuss some core features of Auricle. So in core feature of Auricle, we have live classes. So basically, live classes are for elevating the learning of the students, and in live classes, you will have one-to-one interaction. With the faculty, and then recording sessions are also there. So recording sessions are basically you will have the access of your subject anytime anywhere through the recording sessions. Then also we provide the ultimate comprehensive book with CBME based question answers, MCQ, and full zone for self-assessment, right? Then MCQ is also there from beginner level to expert level, and it consists of standard multiple choice questions. Then also you can book a slot for personalized counseling session in which you will have one-to-one session and then interaction with the counselor. Then we have some subscription plans. So we have three plans: the basic, standard, and premium. So the basic plan is of 3 months, which cost around 15,000, but it includes pre-recorded classes, live classes, and quick revision examination module. In standard 6 months plan, it is cost around 30,000. It includes pre-recorded classes, live doubt clarification, quick revision examination module. And from premium plan that is of 12 months, it's cost around 50,000. You will have access of all the features of Oracle like pre-recorded classes, live doubt clarification, one-to-one personalized guidance, notes, quick revision examination module. And for 20% instant discount, you can use my code that is Aanchal, ANCHAL 20. So as we have started the series of GIT, so today we are having class on small intestine. So talking about the Ma'am, I'm not able to share my screen now. Okay, so as we were talking about the introduction of small intestine, so small intestine is divided into three parts: duodenum, jejunum, and ileum, right? So it is the final, last and final site for major digestion and absorption. So basically, in small intestine, what happens? Complete digestion of food occur here. Okay, so it is the site of complete digestion of food, right? So beside this, small intestine also absorb minerals like calcium and sodium. It also perform or exhibits motility movements like segmental movement and peristalsis. So about segmental movement and peristalsis, we're going to discuss in detail, right? Now, some local mechanism are more important than nervous mechanism in regulation of motility and secretions. And what are those mechanisms we're going to learn? So talking about the length, so as we know, the length of the small intestine is around 6 to 7 meter long. So talking about the particular length of the part, so as we know, the first part of the small intestine is duodenum. Its length is approximately 20 to 30 centimeter. Then jejunum is the second part, length around 2.5 meter, and the ileum is the last part of the small intestine. Its length is 3.5 meter. Talking about the function, so as we know, the first function is the digestion. So basically, the digestion, complete digestion of food occurs here. Then for absorption, some structures are present that are known as villi, which help in the absorption of food. Then secretions are also produced there inside the small intestine, through which the digestion of food complete, like succus centricus. Then motility of food also occurs here, and here also some motility movements also, like segmental movement, peristalsis movement, pendulum movement, and about those movement also we're going to learn. So, as we know that small intestine is a part of GI tract extending between the pyrolic sphincter of stomach and ileocecal wall. So, we know the pyrolic sphincter is present between stomach and small intestine, right? And this ileocecal wall is present between small intestine and large intestine, right? So it is known as small intestine because of its small diameter, and it is longer than large intestine, and length, as we have discussed, it is about 6 meter, right? Now, maximum absorption of digested food products take place in small intestine, and as we have discussed, the proximal part is the duodenum, the middle part is jejunum, and the distal part is ileum, right? Then talking about the some glands, intestinal villi, basically. So as we know, mucous membrane of small intestine is covered by minute projection that are known as villi, right? Now, villi are lined by columnar cells. So there are presence of some columnar cells also, and these columnar cells are known as enterocytes. Now, each enterocytes give rise to hair-like projection, and over those enterocytes, we have presence of microvilli also. This basically increases the surface area of mucous membrane by many folds. So basically, they are present in folds like this, so that the surface area will be increased, so that maximum absorption of food take place here. So as we can see, the simple epithelium cells are present, then capillary are also present, then globular cells, intestinal crypt are also present. So about these structures also we're going to discuss. So, as we know, intestinal gland are simple tubular glands of small intestine, and intestinal gland do not penetrate muscularis mucosa of the intestinal wall, but open into the lumen of intestine between the villi, as we can see here also, right, in this picture. Then the lining of each gland is continuous with epithelial lining of villi. So epithelial cells lining the intestinal villi undergo division by mitosis at a faster rate, right? So newly formed cells push the older cells upward over the lining of villi. And these cells which move to villi are known as enterocytes, as we have discussed. So enterocytes secretes the enzymes, and old enterocytes are continuously shed into lumen along with the enzymes. So talking about in detail, so basically these intestinal glands are the simple tubular glands of intestine. As we have discussed, they open into the lumen of intestine between the villi, and also they contain or consist some different types of cells, right? So first we have the argentefin cells, or they are also known as enterochromaffin cells. So they secrete they secrete a very important substance that is known as serotonin, which help in the stimulation of smooth muscles contraction, so that the motility of food can be occur here. Then the next we have globular cells, which secretes mucus, and mucus is important for providing lubrication motility to the food. Then we have the Paneth cells. So Paneth cells secrete the cytokinin that is known as defensins, and it is important for the antimicrobial activity in the small intestine, right? So this is all about the different types of cells that are present inside the small intestine. Then we have the next one, that is Brunner's gland. So in addition to intestinal gland also, the first part of duodenum contains some mucus glands which are known as Brunner's gland, and these glands penetrate muscularis mucosa and extend up to submucous coat of the intestinal wall, and Brunner gland open into the lumen of intestine directly, and it secretes mucus and some traces of enzymes. So the very important thing that it secretes mucus and also traces of enzyme, right? Then talking about the very important thing that is secreted inside the small intestine, a juice. So basically the name of that juice is succus antricus, right? So secretion from small intestine is known as succus antricus. And talking about the some properties of succus antricus, so we have volume around 1800 ml secreted per day. The reaction and the nature is alkaline, and the pH is approximately 8.3, right? So, talking about the composition part, so as we know, succus antricus contain 99.5% water and 0.5% solids. So, in solids also, in 0.5% solids, we have both organic and inorganic substances. Inorganic, it contains some enzymes. In enzymes also, it has a very good, huge classification like proteolytic enzymes, amylolytic enzymes, and lipolytic. So as the name suggests, these enzymes are important for the digestion of proteins, right? And amylolytic enzymes for the digestion of carbohydrate, starch, and lipolytic enzymes are for the digestion of lipids, right? Now, the proteolytic enzyme, it includes aminopeptidase, dipeptidase, tripeptidates. In myelolytic enzymes, it includes sucrase, maltase, lactase, dextrinase, trehalase. In lipolytic enzymes, it includes a very important enzyme, that is lipase. And along with these enzymes, it also secretes one important substance, that is enterokinase. And about the function of enterokinase, we will learn in the further slides, right? Then other organic substances are also there, like mucus, intrinsic factors, and defenses, right? Mucus is basically for providing the lubrication and motility. Intrinsic factor is basically for absorption of food, mainly vitamin B12, and defenses perform the antimicrobial activity, right? In inorganic substances, it includes sodium, calcium, potassium, bicarbonate, chloride, phosphate, and sulfate. Now, talking about the function of succus antricus. So, the enzyme of succus antricus that act on partially digested food and convert them into final digestive products. So, enzymes are produced and released into succus antricus by the columnar cells, as we have discussed, and the name is enterocytes of the villi, right? Then we have some the name of the enzymes, and we're going to learn what are the substrate over which these enzymes are going to react, and what will be the end product that will be received, right? So first enzyme we have peptidases, then we have sucrase, then we have maltase, then we have lactase, then we have dextrinase, then we have trehalase, then we have the intestinal lipase, right? So the enzyme peptidase is basically for peptidase we have the substrate that is peptides, and the end product we'll receive is amino acid. Then sucrase will act over sucrose, and the end product we'll receive is fructose and glucose. Then we have maltase. The substrate for the enzyme maltase is maltose and maltriose. Then the end product we'll receive is glucose. Then the lactase enzyme, for the lactase, we have the substrate that is lactose, and the end product that will be received, that is galactose and glucose. For dextrinase, we have the substrate dextrin, maltose and maltriose, and the end product we'll receive is glucose. Then for the enzyme trehalase, we have trihalose, and the end product we'll receive is glucose. Then for the intestinal lipase, we have the substrate triglycerides, and the end product we'll receive is fatty acid, right? So basically what happens, talking about the proteolytic enzymes again, if we discuss, so basically these enzymes present in succus centricus and are peptidase in nature, right? And these peptidates convert peptides into amino acids. So basically they are responsible for the conversion of peptides into amino acid, right? Then we have the amylolytic enzymes, right? So amylolytic enzymes of succus centricus, as we have discussed, we have lactase, sucrase, maltase. So convert the disaccharides into two molecules of monosaccharides, right? So here disaccharides will be converted into two molecules of monosaccharides. Now talking about the last one. So for the lipolytic, we have the intestinal lipase that act on triglycerides and convert them into fatty acids, right? Hmm. Now, basically, dietary protein, in continuation of the dietary protein, we have digestion, which includes our luminal and breastwater phase. Both amino acids and tripeptides are absorbed here, and digestion normally quite completed here. Then talking about the dietary carbohydrates, as we know that it serves as an energy and carbo source, and digestion here includes luminal phase and breastwater phase, both are there, and only monosaccharides are appreciably absorbed here, as we have discussed, right? Then for the emulsification of basically for lipid digestion, we have several steps. The first we have emulsification, then digestion, solubilization, and absorption. So basically, physical process take place in stomach. That is for phospholipid protein facility, and in digestion we have the stomach and duodenum, both are involved. And for solubilization requires bile salts, right, which is secreted by liver. And here there will be the role on the formation of micelles. Then there will be absorption of fat and lipid. Fat will occur here, and if more fat are seen in the feces, so that condition is known as steatorrhea. Then we have the protective function. So mucus present in the succus centricus protect the intestinal wall from the acid chyme, right? Thereby it prevent the intestinal ulcer. So basically this is the function of mucus. Then we have talked about the defenses. As we have seen, which cell secretes defenses? The defenses are secreted by this Paneth cell, right? And mucus secreted by globulate cell. So again, as we have talked, the defensins are secreted by Paneth cells of intestinal gland and are the antimicrobial peptides. Then activator function. So as we have discussed the enterokinase also. So here the enterokinase are very important because it activates the enzyme trypsin, and trypsin is responsible for the activation of other enzymes, right? So trypsin in turn activates other enzymes, and also a very small amount of trypsinogen is converted into trypsin by the this enterokinase, and rest of the trypsinogen is converted into trypsin by this own trypsin, right? Then we have another function, that is hemopoietic function. So basically this intrinsic factor of Castle present in the intestine plays an important role in the erythropoiesis because this is responsible and this help in the absorption of vitamin B12 in the terminal ileum part of the small intestine, right? Then we have hydrolytic process. So intestinal juice also help in the enzymatic reactions of digestion. Okay, then we have the regulation of secretion of succus centricus. So for nervous regulation, we study always about the parasympathetic and sympathetic. So basically, as we know that sympathetic is always responsible for decreasing and the parasympathetic is always responsible for increasing the secretions, right? So here also stimulation of parasympathetic nerve causes vasodilation and increases the secretion of secus centricus in the small intestine, and the stimulation of sympathetic nerves causes vasoconstriction and decreases the secretion of succus centricus in the small intestine. Then we all have the role of some hormones also. So basically, when chyme enter the small intestine, intestinal mucosa secretes secretin and cholecystokinin, and both these two hormones are responsible with, and basically they promote the secretion of succus centricus by stimulating the intestinal gland. So basically, both secretin and cholecystokinin helps in increasing the secretion of succus entericus inside the small intestine, right? By stimulating the intestinal glands. Then we have the applied physiology part. So basically, as we know, the malabsorption, right? So as the name suggests, malabsorption is the failure to absorb nutrients such as proteins, carbohydrates, fat, and vitamins. It also affects growth and development of the body. So if malabsorption is occurring inside the body, so what will happen? These substances, which are very essential for the growth and development of body, will not get absorbed, so means simply they will be excreted out of the body. So ultimately the growth and development of the body in the, especially in the children, will get affected, right? Then next we have Crohn's and enteritis. So as the name suggests, this is the inflammation of the intestine, right? So this is the inflammation of intestine known as bowel disease, IBD, characterized by the inflammation of small intestine. Usually it affects the lower part of small intestine, the ileum, right? So in this case, the ileum will be affected and the inflammation causes malabsorption and diarrhea, right? So basically the malabsorption will be there and the condition of diarrhea will be also there. So it develops because of abnormalities of the immune system. The immune system react to a virus or a bacterium that resulting in the inflammation of the intestine, right? So features, we have some features like malabsorption of vitamins, weight loss will be there, abdominal pain, diarrhea, rectal bleeding, anemia. Then we have delayed or stunted growth in children also there, right? Then again, we have another condition that is known as steatorrhea. So it is the condition caused by deficiency of pancreatic lipase resulting in malabsorption of fat, right? So basically in this condition, what will happen if this pancreatic lipase is absent, and basically this enzyme is responsible for the digestion of lipids, for the digestion of fat. So basically if this enzyme is absent, so ultimately it leads to the malabsorption of fat, and this fat is excreted with the feces, right? So in this condition there will be appearance of fat with the feces, right? Then we have celiac disease. So again, the celiac disease is an autoimmune disorder which is characterized by the damage of mucosa and atrophy of villi in small intestine, resulting in impaired digestion and absorption, right? So there will be damage of mucosa and there will be atrophy inside the small intestine. So obviously in this condition also there will be impairment in the digestion and absorption part. Then we have the another very important topic. We're going to discuss some movements of the small intestine, right? So inside small intestine, we have some important movement like mixing movement. Then we have propulsive movement. Then we have peristalsis and fasting, that is migrating motor complex. Then we have movements of villi, right? So for the mixing movement, we have segmentation movement and pendular movement. Okay, so basically mixing means mixing of juices with the food, right? Then propulsive movements, we have peristalsis. So propulsive means to move the food in the anterograde direction, in the forward direction, right? Then in peristalsis and fasting, we have a migrating motor complex. So basically this is a sweeper movement, right? Then the movement of villi, which is responsible for the absorption of food. So in this movement, there will be absorption of food, right? So talking in detail, we have motility of small intestine. So as we have discussed, two basic motility pattern exist. The first we have segmentation, the second we have peristalsis. Okay, so significance of the small intestine serves three functions. The first we have mixing the content, the enzyme and other secretion with the food that help in the digestion. So basically this movement is very important because in segmentation there will be mixing of food with the juices or with the enzyme that are responsible for the digestion of food, right? Then there will be maximizing exposure of the content to the membrane of intestinal cells also, which help in absorption and digestion. Then there is propulsion of content into large intestine. So these are the significance of these movements which occur inside the small intestine, right? Then we have another thing, the segmentation we're going to discuss. So basically why this movement is known as segmentation, because in this the small intestine are divided into some segments, as we can see. So that's why this movement is known as segmentation movement, right? So there will be contractile ring occur every few centimeter, dividing the small intestine into segments, right? So between contracted segments are relaxed area containing small intestine of kind. So as we can see, there will be, if this is a small intestine, so the food will move like this. So if we have food here, so there will be movement of food like this, to and fro. One time it will move forward, another time it will move backward. So basically there will be movement of food like this, as we can see in the shaking of water bottle. So exact movement here occurs here also. And what is the significance? So that this food get mixed with the enzyme and juices which are present inside the small intestine, right? So the segmentation occur about 12 per minute in duodenum and 9 per minute in terminal ileum. Content take 3 to 5 hours to move through the small intestine. So here food will stay approximately from 3 to 5 hours, right? Then we have pendular movement. Why this is known as pendular? Because the movement work exactly like the pendulum, right? To and fro motion, right? One time here, second time here. So in the small intestine also, as we have seen, there will be two to and fro motion of the food, right? So the pendulum movement is the swiping movement of small intestine resembling the movement of pendulum of clock. These are small constrictive wave that move chyme to and fro, up and down, similar to segmentation movement. So both pendular as well as segmentation movement, both are similar and both are responsible for basically the mixing of food with the juices, right? Uh, okay, so now I'm giving one question to the students. I'm giving 30 seconds for this. So secretin and cholecystokinin are digestive enzyme. These are secreted by, you have 30 seconds to answer. Secretin and cholecystokinin are secreted by, this is the question. Yes, the answer is duodenum. Okay, now next question we have the crypts of Lieberkühn, or we call it intestinal glands, are found in gallbladder, intestine, liver, and kidney. These are the option. Again, you have 30 seconds to answer. Yes, intestine. Then the third question we have, the food that enters the intestine from stomach is called as chyme, fundus, chyle, or bolus. 30 seconds I'm giving to answer, everyone. Yes, the answer is chyme. Okay, the last question we have, intestinal enzymes are located on. The options are brush border epithelium, submucosal membrane, lumen of small intestine, or both lumen intestinal wall. Again, you have 30 seconds to answer. Any answer for the last question? Intestinal enzymes are located on. Yes, the answer is D, both lumen and intestinal wall. Now, coming back to the movements. So, for small intestinal motility, we have peristalsis. Basically, here the movement along the tract occurs, right? So, peristalsis contraction are responsible for the forward movement of food, right? So, basically, what happened? There will be contraction and relaxations of the muscle fiber so that the food can be moved in the forward direction. So, in time zero, so there will be contraction in this part, so the food will move in the forward direction. Then in seconds later, what will happen? There will be relaxation of this part, and again the contraction or constriction of this part will start. Then again the bolus will move in the forward direction, right? So as we can see, there will be contraction in the longitudinal and circular muscle fiber, and in this way, there will be movement of food from mouth to anus. So in the whole GIT, there will be contraction and relaxation of the muscle fiber occur, so that the movement of food, forward movement of food can be occurred. So in the first step, as we can see, there will be contraction of circular muscles behind food masses. Then there will be contraction of longitudinal muscles ahead of food masses. So in this way, the food will move in the forward direction. And in third step, there will be contraction of circular muscle layer forces food mass to the forward side, right? So then next we have MMC. So in the last class also we have discussed about the MMC. So basically, this is peristalsis in fasting. So this type of peristaltic contraction, which occurs in stomach and small intestine during the period of fasting for several hours, a large portion of small intestine or intestine is involved in this contraction. And here the velocity for the traveling is approximately 6 to 12 centimeter per minute, right? And it sweeps the excess digestive secretion into the colon and prevents its accumulation in small intestine and in stomach, right? So basically, it sweeps the residual indigested material into the colon so that defecation process can be initiated, right? Then talking about the movement, so as we have talked, the movement of villi are shortening and elongation. So here it occurs alternatively in helping imprinting lymph from the central epithelial into lymphatic system. Here the surface area of villi is increased during this elongation, and these movements are caused by local nervous reflexes which are initiated in the presence of enzyme, in the presence of chyme. So basically whenever food come inside the small intestine, it get initiated, and villi is another hormone which is believed to play an important role in increasing the movement of villi. So basically, with the help or with the movement of villi, there will be increase in the absorption rate of the food also, right? So in last, basically talking about the summary. So small intestine is the last and final digestion and absorption foodstuffs occur here. The intestinal juice known as succus centricus, the enzyme of intestine are embedded in the wall itself, not released into the GI lumen. Then the monosaccharides, amino acids, and short chain fatty acids are absorbed in blood, whereas long fatty acids are directly absorbed inside the in lymphatics. Then as we have talked about the functions of small intestine already, now we are going to see a summary and conclusion. So as we have discussed here, the mechanical movement of small intestine occurs, which help in mixing of chyme with the digestive juices like succus centricus and pancreatic juice and bile. Then we have secretory function. So basically, small intestine secretes succus centricus, enterokinase, and GI hormone also. Then another thing, we have the hormonal function. So as we know, the small intestine secretes many GI hormone like secretin, cholecystokinin, and these hormones regulate the movement of GI tract and secretory activity of small intestine and pancreas. And again, we have seen the digestive activation function, hemopoietic function, hydrolytic function we have seen, and absorptive function. We have the presence of villi and microvilli in small intestinal mucosa, increases the surface area of mucosa, and this facilitates the absorptive function of small intestine, right? So here protein, carbohydrates, and fat and other nutritive substances such as vitamins, mineral, and water are absorbed mostly in small intestine, and from here these substances enter the blood directly or through the lymphatics. So for the university exams, the important questions are composition and function of succus centricus and discuss the regulation of its secretion. And another question could be the discuss the action of intestinal enzyme that we have already discussed in the slides. Okay, so this is all about the small intestine. Again, I'm repeating the important question. All the movements are very important, right? In small intestine, the function of small intestine are important. The succus, the composition, property, and function of succus centricus are very important. The question over this topic can also arise in the university exams. Then intestinal glands are important. Yes, and function of small intestine is also another important topic. So this is all about the function and structure and the important content which is present inside the small intestine. Thank you so much.